That's Bully up there. We're the Bulldogs. He was in the hospital the other day because he's the Mississippi Foundation sire for Bulldogs. I didn't even know it was a dog. I walked right by him, but he was in there. Getting ready for this talk, one of the things that surprised me is we've only known the rotavirus as a cause of diarrhea in foals since '75, which is really pretty recent to me. And even in kids, it goes back to 1978. So this is, to me, a fairly recent pathogen. It was first seen in the United Kingdom in a diarrheic foal found with electron microscopy. So we're still pretty early on that. Is this the clicker? Sorry, it's my joke slide. I messed it up. When I was also putting this talk together, looking at all these different rotaviruses, I just wanted to get a list of what all rotavirus can be affected by rotaviruses, and it reminded me of like the Tasmanian Devil's menu in the cartoons where they say, what does a Tasmanian devil eat? Well, he pretty much eats everything. And rotavirus does too. The A's and B's are the ones we're going to talk about today. But before in horses, if you look at papers before what's going to be published from this study, everything said only A's had been reported in horses, and these other species had B's. And if you look at B also, and a lot of these other species, that disease can be subclinical or really mild. So to me, it's interesting that in this outbreak, bees tend to cause such severe disease in these foals. So anywhere from 2 days, sometimes even earlier than that, I've seen them, to 6 months is kind of our range we use for what age range is affected by rotavirus. Incubation period can be as short as a few hours, but generally 1 to 4 days. And it can be asymptomatically shed by both mares and foals. So there's been studies where they could find rotavirus being shed by mares with foals that were affected, and other studies that showed differently, but can be shed asymptomatically. Generally, a lot of them are affected, but very few of them will die. And most of our clinical signs that we see are due to dehydration. So I always feel like in these foals, if I can prevent secondary problems, then I have a pretty good chance of getting them through it. Foals on average will shed this virus for about 3 days after the resolution of clinical signs. That can go up to 12 days, and that's true for asymptomatic foals as well. So asymptomatic foals can still shed this virus for the same period of time as symptomatic foals. And I'm definitely out of my lane talking about this. So like I always tell the students, if somebody else tells you different, I'm the one that's wrong. But we're talking a little bit about the genetics of these viruses. Is these different antigens are where our tests are based, and trying to get vaccines, trying to go after some of these sites that are more likely to be the sites of neutralizing antibody is where we have to go. So how our tests kind of let us down, and didn't really let us down in this outbreak, but just weren't able to detect these new antigens, and that's where some of this got biased. So go through some of the surveys that have been done in Ireland and Britain, and this was looking at foals that had diarrhea. About 37% of those foals had rotaviral diarrhea, again reported in that paper. None of those rotaviruses were identified other than the type A. And also in the United Kingdom, rotavirus in 24% of foals with diarrhea. And in that study, they also looked at controls on farms. These were unaffected foals. And so found that some of those unaffected foals also were shedding rotavirus. So when you added those foals in with all the affected and unaffected with diarrhea foals, 17% of those foals were found to be shedding rotavirus. A study done in Florida, and these were foals, but it was under 10 months of age, that were in the hospital. They either came in for diarrhea or developed diarrhea within 48 hours of coming into the hospital. About 20% of those foals were isolated with rotavirus. And interesting to me that over 1 month of age, which to me is usually I tend to think of rotavirus these younger foals, but found it more in foals over 1 month of age. It's work Dr. Slovis did looking at PCR in Kentucky. So PCR looks for DNA. We'll have a slide of that here a little bit later on, and found that 35% of the foals that they looked at were with diarrhea had rotavirus. Again, also looked at foals that were unaffected, and only 3% of the unaffected foals, the healthy controls, shed rotavirus. And interesting out of this paper was that these secondary infections, because that's important because rotavirus kind of opens the door. It damages, we'll talk about this a little later, rotavirus damages the lining of the intestine, so it opens the door for some of these other pathogens to occur as well. And that 61% of those foals had some other pathogen, and one of the more common ones was Cryptosporidium, which is a protozoa, which is pretty uncommon cause of diarrhea. So interesting that there can be more than one thing. There can even be more than one strain of rotavirus that affects the foal. So it may not necessarily just be a single strain. It may be different antigenic strains within the foal. So, and also getting ready for this talk, one of the things maybe look into this is like, well, where did this stuff potentially come from now that we have this B type? And there have been reports, sporadic reports of rotaviruses when they were typed out and genetically analyzed that had genetic similarity with bovine and porcine rotaviruses, like some of those B rotaviruses. In Argentina, there have been cases that were genetically similar to canine and feline rotaviruses. And it's believed those are just trans-species infections that occur. The genetic reassortment would be where two different rotaviruses are going to infect the same cell at the same time, and they can do some DNA sharing. That's believed to be pretty uncommon. It's believed that we just are having some cross-species transmission for these other strains of affected foals. And rotaviruses is resistant to many of the commonly used disinfectants that we talked about. Can stay 9 months in the environment at room temperature, so it hangs around a while, so it could easily survive to the next foaling season. And we talked about fomites, especially the two-footed fomites that spread these things around the farms, are important. And at least in pigs, 1 ml of feces can contain, 1,000 million, 10 billion viral particles can be in 1 ml of feces. And as few as, this is also pigs, as few as 90 viral particles can infect a naive animal. So knock 2 zeros off that number, and that's how many animals you can infect with a single ml of feces. So kind of what does it do? It affects the lining cells in the intestine and takes off the cells on the villi. The villi are like the little fingers that increase the surface area there in the gut. And the digestive enzymes are on the end of those fingers, so the things that help us digest proteins and digest sugars are there. And also that's where most of the absorption occurs. So it both takes out the ability to digest the nutrients and to absorb the nutrients. And when those sugars are able to stay out to the lumen of the gut, they attract water and they hold water and they can be fermented. So just in this diagram here, just have the lactose molecule there at the top. Normally lactase comes out, it's going to break that down into glucose and galactose, two simple sugars that gets absorbed. But when rotavirus over there on the right affects the cell, kills those lining cells, so it interferes with both the digestion and the absorption. And normally that crypt there down at the bottom, that's unaffected by rotavirus. That's where those new cells will come from. So they have to come from that crypt, crawl all the way up around the top of that villus to repopulate, and that takes at least 3 days. So once rotavirus has infected the villi, taken out the end of the villi, so taken out those cells on the end of it, where the absorption and the digestive enzymes come from. And so we shorten them. They're blunted. I have a picture of this coming up. If we have less lactase, the big enzyme there in the top right, lactase is going to be normally secreted, go out and break down the lactose. That'll be absorbed. So there's less lactase, so digestion doesn't occur. That nutrients stay in the gut, hold water. Sugars get fermented by bacteria, get produced. Usually acids are produced, so those can be irritating directly to the gut. Normally water is going to be absorbed also, but when those lining cells are gone, the ability to absorb water from the gut is going to be reduced. There's more bulk in the gut as well. And there is some evidence that there may be some secretion, a secretory diarrhea that's caused by rotavirus. And rotavirus may also have either direct effects on the motility of the gut, like Dr. Brown was talking about earlier, that problems like ulcerations and intussusceptions have been associated with rotavirus. It hasn't been proven, but they've been sure associated with that. And things like intussusceptions as well. So whether rotavirus, either through irritation of the gut or actually a stimulation of the enteric nervous system of the gut, does affect motility, is at least speculated. In this picture, it's showing what the gut would normally look like with those large villi, and then those blunted villi. And one of the problems about being in academia is once you double the students in age, you can't tell them anything that they associate anymore. So I don't even know if they know what shag carpet is. But to me, I thought, there's shag carpets, what it normally should look like. I haven't asked them. They probably don't, though. So you have shag carpet, rotavirus goes by, kills those villi, it flattens it. You can see there's just less surface area. So we lose the enzymes, we lose that area to absorb all these things back. And also, if there's effects on motility, that ingesta is going to move through there faster. So a little bit on diagnosis, where we've used electron microscopy to look for these viral particles. We can do polymerase chain reaction, which looks for DNA. We can also use the human assays because there's some similarities between the human and the animal rotaviruses. And also, they can be co-infectious. Can be those other infections that are present there as well. So Clostridia, which was found in this outbreak, Clostridia can be there. Salmonella can be there for sure. With stress, the gut's more likely that Salmonella can be into the gut because the gut's damaged, so it kind of opens the door. And Cryptosporidium is found in Dr. Slovis' study. So electron microscopy, it's a specialized technique. You have to have an electron microscope. You have to look really close for those particles, and you have to have a certain number of those that are present. So if there's not a lot of them, you can miss that. So that's a really specialized technique. PCR has really revolutionized our ability to work and to diagnose things. Again, something else students take for granted is how hard we had to work before we had these tests to figure out some things. PCR looks for DNA. So really the way this works, if you heat up DNA, it unzips itself. You can put in the things DNA needs to put itself back together, the nucleotides and enzyme that helps that occur. And each time you heat it, you get more copies. So it's exponential. So 1 becomes 2, 2 becomes 4. So that way we can amplify what DNA is present. But we have to have a primer, so we have to know what gene we're looking for to kind of get that reaction started. So if we don't have the right primer, then we're not going to find it. And that was one of the ways that rotavirus type B got away from us, was because we're using the type A primer, because we, before this year, didn't know that anything other than A affected these horses. The other thing are these detection levels of PCR. And so the more DNA that's there, the faster you'll find it. So that's why it's important. Sometimes your veterinarian will talk to you about the CT times. It just means how many cycles it took to be able to find that DNA. So if you find it faster, it's less cycles than if there's less DNA. Antigen detection kits, there's kind of one of our fast cornerstones in the hospital, because these only take about 15 minutes to run. They work on this VP6 protein, but that protein is where we classify rotaviruses into their ABC group. So again, we're looking for that protein in an A and not the B. So that was one of the reasons why it was missed on this test. The B was missed on the current test. Dr. Slovis's work showed that the PCR test had a poor correlation with those rapid tests. Other studies have found with electron microscopy there was good correlation. So if we had negatives on these rapid tests, again, it may take 3 negatives to really rule it out. If you get a positive, it's pretty good information, but because it's negative on the rapid test, it may not, doesn't mean it's not there. In the AEP guidelines, everybody's familiar with this. It's always going to be the same for mare, whether she's previously vaccinated or not. The foal has to get colostrum and get enough colostrum to have a shot for these antibodies to be protected. Just a review of what's out there. There's, as far as I know, 3 different rotaviral vaccines that are commercially available, and they go after different antigens on the outside of the rotavirus capsid. And the neutralizing antibodies are pretty much against that VP4 and VP7 protein. So those are where the antibodies tend to be directed against. And you see in Argentina, there's a trivalent vaccine that's available. Here in the UP and in the US, we've got monovalent, and also in Japan. At least in nature, it appears that those G3Ps and the G14Ps are the most common strains of rotavirus. There's been some work here in Kentucky. The diagnostic lab showed that G314 is our most common one, but more or less in the world, in the United States, we're dealing with 2 different strains of the rotavirus. Foals and the mare can all have infected high titers and still become infected. So even though we can get high immunity in the mare, foal ingests enough colostrum, still gets enough high antibody titer, they can still become infected. In studies that have been done, they've shown a reduction in diarrhea in the falls. One other study showed it was reduced, but not significantly reduced. So, in all, vaccine's pretty efficacious, even though we have breaks. It does produce good antibody, and it does reduce the incidence of rotavirus on the farm. To me, it's interesting, and maybe this is something we can try to chase next year, is at least in calves and lambs, it's been shown that continued lactogenic immunity, continued antibodies that are in the milk, have a large role in preventing infection. So the window's open 12 hours, we're supposed to absorb the colostral antibody. But if we have some continued immunity, at least in other species, that's been shown to reduce the infection. So maybe that's something we can chase next year. Again, all three vaccines, both the monovalents and the trivalent, have been shown to have good antibody response. Also, they've gone back and looked at the mare's response to other types of rotavirus and shown that she can have good response even to ones that she hadn't been exposed to. So even though we have monovalent vaccines here in the United States and also in Japan, it's been shown that they still get a pretty good response to any of the types. So it does work well to vaccinate them against these other strains. But again, this should be considered to be partial immunity, that other things can happen, probably infectious dose if they get enough virus, because just a mill of virus, at least porcine, can be a huge amount of virus that they can be exposed to, the level of their colostral immunity, and probably other virulence factors. Because some of these rotaviruses, even if they go through digestion, like with trypsin, that can make some of these viruses be more infectious. So kind of where do we go from here? The G14P in Kentucky has been shown to be with the primary strain, and that's been some work done here at the diagnostic lab, along with the GP3, other one. Those are the two most common ones that circulate. So, that would be a good candidate. There's some work in Japan to show that is a good vaccine antigen that we should add in to help protect against other strains. To see what happens, does A kind of go away, or do we become more B next year? I think that'll be something we'll have to figure out also. Herd immunity, maybe it'll do us some good. It seems like sometimes it does in these outbreaks. And there's only been a few good things come from COVID, but I guess everybody knows about biosecurity now a whole lot more than they used to, and everybody knows a lot more about vaccines than they used to. So at least client education, I was thinking, well, at least we got that for us, that client education is going to be easier on this. And finally, I think it's really important to recognize the work that everybody in this room has done. You think about, West Nile popped up within a pretty short amount of time, got the vaccine that worked really great for that. COVID popped up. Hopefully that vaccine's going to work. And in one foliage season, people in this room figured out what the problem was, made a brand new test, and hopefully we're going to get somewhere to reduce this. So I think it's remarkable what people have been able to do. And finally, for that farm manager or veterinarian that has everything, you can order them a rotavirus off of Amazon so they can keep it by the barn door and punch it as you walk in. So it popped up on my searches as I was stealing images off the internet, but it was on there. So, are we taking questions or no? Okay. All right. Thank you.